Hello there, and welcome to this Khan Academy tutorial on A-level biology. In this video, we will look at receptors, membrane potentials, and action potentials. So let's get started. Here are the aims of this tutorial. We will explain the function of a receptor, describe how the resting membrane potential is maintained, define threshold potential and action potential, describe how an action potential is generated, and explain what is a refractory period and its importance. So let's start by understanding what receptors are and the types present in the body. Receptors of the nervous system detect a stimulus, which is a change in the environment. The receptors can generate an action potential when a threshold is reached. We'll discuss about this later in this video. There are many different types of receptors, but the key ones are mechanoreceptors, chemoreceptors, and photoreceptors. Mechanoreceptors detect pressure, and an example of this is the Pacinian corpuscle. Chemoreceptors detect change in pH and other ions, and photoreceptors detect light. Examples include rod cells and cone cells of the retina. Now let's look at membrane potential. This will seem confusing and difficult to understand at first, so I will run through it step by step. You see, in our body, certain ions are of different concentration inside and outside the cell because of the selective permeable membrane. This generates a potential because there is a concentration gradient for the ions, meaning a difference in charge inside and outside the cell. And so, if the cell permeability changes, the membrane potential changes. 4 key ions to be familiar with at A-level biology are potassium, sodium, calcium, and chloride. Generally, we have a higher concentration of potassium inside the cell and a higher concentration of sodium outside the cell. The concentration of calcium and chloride ions are important in transmission of electrical impulses across the synapses. Okay, hopefully that made sense. When a cell is at rest, the voltage it carries is called the resting membrane potential. Now you may be thinking how I added the word voltage out of nowhere. But hear me out. Voltage and potential are interchangeable terms here. I agree this is not fully true, but for A-level biology, accepting this would not make you lose marks in exams. At rest, the cell is at minus 70 millivolts, and it's more permeable to potassium ions than sodium ions, as many sodium ion channels are closed. A common exam question asked is how is this resting membrane potential maintained? And the classic mark scheme answer is as follows. The 3 sodium ions are actively pumped out of the cell in exchange for 2 potassium ions. This increases potassium ion concentration inside the cell. And some of these potassium ions diffuse out of the cell via potassium channels down an electrochemical gradient. This will be discussed in the next slide. Note that this process is an aerobic process, and so it requires energy in the form of ATP. So let's answer the question of how the action potential is generated. Well, you have to reach the threshold potential first. This is the level needed to generate an action potential, and for our cells, this is approximately minus 50 millivolts. A point to note is that all stimuli can generate a potential, that is, increase the voltage by increasing the membrane permeability to sodium ions, so that they can enter the cell. This makes the membrane potential less negative, that is, from minus 70 to minus 50, as sodium ions are positive. And if a stimulus causes enough sodium ions to enter the cell and reach the threshold, an action potential is generated. You can see that some stimuli fail to generate an action potential as their intensity is low, meaning not enough sodium ions entered the cell. Okay, now let's look at the generation of this action potential. This is complex, so do take some time in taking this information in. Don't be put off by this diagram, it will make sense as I'll go through it step by step. So once threshold is reached, a special type of channel protein called voltage-gated sodium channels, VGSC for short, open, and this causes a rapid influx of sodium ions into the cell, causing it to depolarize. The word influx just means moving inside. The word depolarization means making the membrane potential positive, because the word de means opposite. Since a cell at rest is negative, it becomes positive when depolarized. Once the cell reaches about 40 millivolts, don't worry about remembering this number, the voltage-gated sodium ion channels close and potassium channels open to restore the membrane potential. This is called repolarization, i.e., restoring the cell to its negative state. Unfortunately, our cells are not brilliant at controlling how much potassium goes out of the channel, and so a lot more potassium goes out of the cell than needed. This causes the cell to hyperpolarize. Hyperpolarize is where the membrane potential reaches below minus 70 millivolts, that is lower than the resting potential. To counteract this, potassium ion channels close and the sodium and potassium ATPase becomes activated to restore the membrane potential back to normal. When a cell is hyperpolarized, it enters a refractory period. Here, no action potentials can be generated no matter the intensity of the stimulus. The word intensity refers to how powerful the stimulus is. Imagine the difference between being pinched and touched. This occurs because voltage-gated sodium ion channels can't open, and the benefit of this is that action potentials can't overlap, and it also allows action potentials to travel unidirectionally, that is, in one direction. So here's a recap of the aims we went through in this tutorial. As usual, use them as questions to consolidate your learning. Thank you for watching, and I will see you in the next one.